Well, I don't really need them. We'll show our video at the very end, I guess. So I don't really need them. Not right now. Okay, it's 4 o'clock, right? Are you good?
Yeah.
Is everything good?
Yeah.
What do we do with my phone?
You gotta talk loud because of the fan.
Ladies and gentlemen, good evening. I'm going to start right on time, unusual for us here. My name is Lisa Karn. Happy New Year. We have to be very punctual today because yoga is happening in this room. Normally at 6, we ask you to move to 6:30, and we have a lot of information to share. So I'm going to start right on time. I'm going to go first with an overview and history of coral reef restoration in Belize, because what you are invited to tonight is part of our annual training course. But what makes it so special this year is that we have 3 invited guests with us. And I'm going to introduce them now, and then I'm expecting more people will straggle them later. So when we get to their talks, I'll have them probably just introduce themselves. But Dr. David Vaughn is here. He is very famous used to be at Mote Marine Laboratory, very famous for microfragmenting. He now has an organization, Plant a Million Corals. This is his 3rd trip to Belize, so we've been working together for a few years already. We also have Dr. Mark Butler with us. First time in Placencia, but been working in Belize about 30 years. Originally lobsters, moving into crabs, some other good stuff. I'll let him, again, when your talk is up, you can get a quick sentence from Old Dominion University, which is in Virginia. And with him as well is Jason Spadero. And Jason I first met, I met Mark ages ago, he doesn't remember. And then I met Jason at a couple of regional conferences. And why we're so excited about having him here is we've been excited about crabs and their ecological role on the reef. And these are the crab guys doing all this amazing work with all this amazing data, so we don't have to. So we can hopefully start moving into thinking about ways that we can restock the crabs we like to eat, but also we'll be talking about some other species of crabs as well. So again, you're invited here because of our special guests, but this is part of a 5-day annual restoration course that we've been doing since 2015. We have a lot of funders at the end of my talk show. You'll see all of our different funders, but this week specifically is being funded by WWF and MCAP. MCAP stands for Marine Conservation and Climate Change Adaptation Program. Don't laugh, it's always hard to get all the acronyms straight. With that, am I going loud enough for you, Maya? I'm going right into it. This photograph that you were seeing was natural aquaphores at Turnef. So this talk is a little biased towards Turnef because I just did a funder presentation in October, so then I updated it with all the things I wanted to share. It's probably too small for you to read. This is for the course participants. We'll be going over the agenda later. You know what you're doing tonight. If you're part of the course, you should be checking in with Monique. You should have signed up with her. You should know where you're staying. You should know what you're doing. tomorrow, we're all meeting at Seahorse Dock at 9 a.m. It's snorkel only. Wednesday, 9 a.m. Seahorse Dock, diving potentially. Thursday, 9 o'clock Seahorse Dock, diving potentially. And lunch is provided at sea each day. So that's just a quick, I know half of you here are not in the course, but that's just a quick rundown on our logistics. So hopefully, many of you should know this already. We're speaking about the critically endangered aquatic corals. In Belize, we call them elkhorn, staghorn, pipe shank, pantail are the Creole names, and there is a hybrid between the two, which they call fused staghorn or Aquacora prolifera. And so you're going to read, hopefully, can you guys all see the slideshow good enough? Is it bright enough? Is the font big enough?
Maybe that window over there, one closest to the screen, that might be good.
Because typically I don't read the slides, I let you guys read the slides, and then I can just ramble away as usual. So, if you don't know, it's staghorn on top and elkhorn on the bottom in this photograph. The main causes for loss, again, they are adapted to high energy, but because of climate change, we're getting obviously more frequent and more intense hurricanes. Then bleaching, disease are both related to climate change because it's affected by the higher sea temperatures. And predation, predation means things that eat things, right? So most people don't realize there are things out there that eat corals. Corals are actually live animals, and it turns out there's a lot of things that eat the corals, and the things that eat the corals are eaten by the things that we like to eat, like lobsters and triggerfish and things like that. So predation is actually another issue if you have a lower population of these endangered corals, and then your ecosystem is out of balance because it's over-harvested. Predation can actually be detrimental as well. This is an example of a snail. These snails can, you can turn it over and they always have the orange foot, or orbiculum, or orbiculum. Like on the conch is the foot, right? It closes on the snail, and so the ones that eat the corals, it's bright orange. So that's how we know that's the bad one. And they've also been shown to spread disease. So if a coral has a disease and a snail, or even potentially the fireworm is eating that coral and moves to another coral, it can spread the disease. So that's why it's also a serious problem. I hope you can read the font. If not, I'll just tell you that there's a 10-year-old economic study for Belize specifically that was done by World Resources Institute, and it valued just the reef ecosystem services, shoreline protection, habitats, and aesthetics or tourism value at over almost $400 million a year in Belize, and that's a 10-year-old study. And so last year, WWF did a specific study on just Laughing River Key National Park, just 12 or 13 miles from us in Placencia, and that marine protected area alone was valued at $4.5 million a year. So there's a lot of reason to protect our reefs, right? This one I like to show for several reasons. Let's see if this plays. Erosion is a serious problem that all of southern Belize, in fact in the Caribbean, all over is happening. This video is from a few years ago before. Stop it, stop it. Before we moved the restroom. So it was a serious problem because it was actually affecting the restroom at Laughing Bird Key National Park, which of course we don't want wastewater into the areas where we're planting corals. The other reasons I show these photographs, the top black and white one is an actual photograph from the 1970s near Placencia, and it's showing that both the elkhorn and the staghorn live right next to each other. And I also show it because the bottom photograph is nursery grown outplanted corals at Laughing Bird Key. And I've been doing this for well over 10 years, and Dave can back me up. One of the criticisms was you can never restore back to what it was, right? So what are you aiming for? So I show this all the time to illustrate that, in fact, and you'll see in the talk, we can bring back the live coral cover to a very high amount, but we can't do it everywhere. So we have a lot of site selection tools that we have to be smart about where we put our time and energy into which sites we're going to restore. The other reason I show it is because people were even critical at the time about mixing up the aquifers and insisted that elkhorn lived in one zone and staghorn lived in another zone. And obviously that is not true, right? So our strategy to date, if you can't tell with the naked eye, those are two different staghorn genotypes. For those of you that don't know what a genotype is a different individual So when I say different genets, it's two different individuals. So in the bottom left, you have elkhorn there. You can see they have different growth forms, but also more importantly, they have different patterns and resilience to bleaching. Bleaching is caused by higher temperatures. And on the bottom, we have staghorn with the prolifera. So the strategy is that we put these different individuals in close proximity together so that when they do spawn, it's easier for them to cross-fertilize to meet another individual and make more baby corals and reseed the reef. So we're just sort of boosting nature. And we have been able to document our nursery-grown out-planted corals spawning 2014 every year through 2017. We didn't check 2018. So we're excited about that. So the mapping. So this little map here is just staghorn, and the green means giant sands over 50 meters in my direction. Course participants will see some of this tomorrow. The yellow means scattered here and there. I'm explaining our strategy. So we map where the existing corals are. We do the mapping during the hottest months. We're looking for the healthiest, strongest corals to then propagate in the nurseries and outplant together. In this map of Saltwater Pea Reserve, the colors mean different things. They're representing the three different apicorid taxa, the staghorn, the elkhorn, and the hybrid. And the photograph is natural staghorn in very shallow water in our hottest months. So this is what I mean when we source and we map. We're looking in our hottest months, we're looking for the corals that are not bleaching, that are not diseased, and those are the ones we're trying to propagate and put together and put out there to make more baby corals that can stand the hotter temperatures. So again, a little bias towards Turneff Atoll Marine Reserve because it was my most recent PowerPoint that I could just update. And maybe you can't see from far away, but very close to each other we can have a completely dead reef and then a live reef. And a completely not so good looking staghorn and then this huge patch of staghorn. And we still don't have the answers to this why. It's not strictly genotype, it's not strictly the microenvironment. What's going on? There's a lot of questions. Last September with Mar Fund, we finally got Dr. Ileana Baum from here, who is a very famous, sort of the premier apicorid and coral geneticist. And so she finally got to come and see all of our work. She didn't even have the answer yet. So there's still a lot of questions out there. The long story short, though, and I'm not going to read through this, is that we have a long list of site selection criteria. And we adhere to that because there's no reason, for example, if you have a very turbid, very polluted area or a place where there was no corals, you would not invest your time and money in putting corals there. But believe it or not, we get those questions all the time. Can I put a reef in front of my resort so I don't have to drive the boat far? No, you cannot if there was no reef there, right? So this is in our manual, and all of our participants that are here have our updated version of the manual. But our old version, soon to be replaced by the new version, is available on our website, fragmentsofbook.org, technical documents, media, something on the far right. So the old version is up there right now, and the new version just came out, will soon be up there as well, because the new version includes our results so far with Dr. David Vaughn and the microfragmenting. And this video is showing you why the MPAs are so important, because we need those fish to help us keep the algae down. The angelfish eat hydras, the sponge, and the parrotfish, of course, are eating the algae. So we just got some results from Dr. Claire Paris, and I don't have it all to show you, but the idea behind what she was doing for us is adding to our site selection criteria. In Belize, we are still blessed compared to other Caribbean nations, and specifically Florida. We have a lot more healthy corals in a lot of other places, but as I mentioned before, they're in patchy areas. So for over 10 years, we've been mapping where these corals are. All the photographs in my talk are from Belize, right? So we map all of these, and we give them to this expert in University of Miami, and she can use the wind patterns, the current patterns, the benthic, meaning what kind of substrate we have on the bottom. Is it seagrass? Is it reef? Is it someplace the coral will settle? And then we've been doing the spawning, so we know when the corals spawn, we know sort of how long they float around until they land, and she can make a computer model sort of simulating where we're expecting our naturally healthy reefs to be reseeding themselves. Why? So then we can ground truth, see if that's so, and if that's so, don't bother going there. Let's fill in the gaps. Let's go restore and put corals where they're not getting reseeded already. I hope that's clear. If it's not, you can ask me about it afterwards. Normally I let people ask all along, but like I said, we're restricted to time. That's why I'm talking so fast, because we have a lot of people to share information. site. So this is just an old example of mapping of the aquaporids. And then we've done multiple consultations over the last 10 years, but specifically under the IMPACT program when we moved up to Turnef, again biased towards Turnef, so I'm showing Fisher's consultation in Chinook, very northern village. And what we would do, and I forgot to bring the map, is that we would bring the map out of the atoll and we would ask the fishers and the tour guides, where do you know where these corals are? Because those guys are in the water all the time, right? And so since day one, that's how we've gotten a lot of our information by working with the local tour guides and fishers that have that information. This is an example of the map. We would just put it on the wall and they would mark out and we could go out there in ground truth and actually see if those corals were there. So this is an example of what Claire Paris was doing using the wind patterns and things like that. The benthic information, which by the way needs to be updated. Our benthic maps, anybody listening, have not been updated since 2005. So that is something we would like to see updated as well. Then all the hydrodynamic data for the spawning. And my fancy little video of the spawning didn't work, but what this is, she just gave me all this information, and it's a whole lot, it could be a whole talk by itself. But this is what it's representing as connectivity. So when all the corals spawn, the circles and the size and the darkness mean that that's more important connectivity areas. So those are the areas that are actually, producing all these different larvae. And I'm sorry I can't get into more detail with that because there's so much to talk about. Do I have a video here? This is just a quick map of Belize and all the areas we've been working and that we've been doing this for over a decade now. I want to give you a quick reminder why we started. It was Hurricane Iris in 2001, Category 4. It hit Laufenburg Key directly and Placencia as well. So we were looking at lone dead coral and rubble everywhere. And now what you're looking at is nursery-bound outplanted corals at Laufenburg Key National Park. So this is just a little snapshot to bring you back into the big picture, what we're doing, why we're doing it. Methods. There's lots of ways to grow corals. I don't want to stress on that so much. What I want to stress about later is the outplanting. And these are old photographs, examples. For example, Linda Garcia planted that. That's the prolifera. That's the hybrid that I mentioned in the far corner over there. These were some photographs, and even there's Annalise, who always comes to our meetings too, from 2010. So this is an old slide. Then we also adapted. We had an earthquake in 2009, and we lost these cliffs underwater of reef, all the patch reefs around the fringing keys. And so where are you going to outplant the corals? Because typically we plant on old dead coral heads. How can we do that? So we borrowed from this overseas place called Seascapers, where they were putting multiple species on each frame, and instead we put a single genet, where a genet is an individual of each species, on that dome, again with the idea that when they do spawn, their gametes can mix. So these particular corals are hermaphrodites, so they have both male and female. So when the bundle comes out, it has both inside, but it can't self-fertilize. It needs to be another coral. So this is what I keep trying to emphasize, that we have to have the different genets or different individuals of each species in proximity to each other, so that when they spawn, it's easier for them to make more baby corals. And we've done this in other areas, and we put it on the old rubble. The other thing the earthquake did was make it very clear to anyone who never believed this staghorn used to be everywhere. Like all the older fishermen told me they used to be annoyed because they couldn't move their boat around. There was so much staghorn. So when we had the earthquake, and there's still sites you can see just this sheer drop, and all you see is that dead rubble staghorn. It's very obvious it used to be everywhere. So super new since 2017 is working with Dr. David Vaughn, and we did some size class experiments. He came here first in March 2017, and we basically, remember, we're not working, we have no land-based nurseries, no aquariums, no tanks. Everything we do is in the sea. And so we have decided that in the natural environment, we're doing a little bit larger. Maybe we could come up with a new term, micro, macro, I don't know. Because we're not doing as small as he does, like I want to call it from 1 centimeter. We're doing more or less like 5-centimeter pieces. But so far, we're so thrilled with it. These are some time series photographs where we did not put these in the nurseries at all, just cut them up, put them directly on the reef at Saltwater Key. You can see, remember I mentioned the genets, this was the same genet or individual, so they grow right into each other. And then starting to make a nice big colony by themselves. Super exciting for us. We time we could step this process up and make it go even faster. And we're already getting fast results. This is a natural picture of a coral that we sourced when Dave was here. We took off the branch to do the microfragmenting, and I hope you can see with the naked eye, this rim inside there was this coral, that arm was actually dead, and it grew over its own self. It resheeted its own self, and you can see how fast it has grown back. So my point with this slide is that not only do we need to be smart about where we choose to outplant, we have to be smart about which corals we choose to propagate, right? And so when I mentioned we're looking for the stronger, faster growing ones, this is a perfect example of, wow, this coral is like, it's really growing like crazy, it can self-repair, this is what we want to propagate and do. And so we've been evolving even in this short period. So Dave brought us the saw that he works with the really, really small corals, and then University of North Carolina was here, and they left us with a larger wet tile saw, but we still use the diamond blades, and this Franco and others prefer because we're doing larger pieces than Dave's been doing on shore. So he can work much quicker with the larger fragments and making larger pieces. Those of you in the course will be taking both saws out on Wednesday, so you'll get a chance to use both of them. So this is an example. These are some other species. This is, for those that didn't know, Psittacea and Strigosa, a star coral and a brain coral. And this is the University of North Carolina's work. And it's just another example of collaboration. And they're not quite microfragments. The idea is it's a long-term experiment. They're looking at nearshore, offshore growth rates. And when they're done with their growth rates, we will take those corals, take them off that disc, because it's artificial, and then reskin and do some experiments with reskinning on some old dead coral heads. We've also transitioned from the beginning with Dave. We were using little broken bits of the cookies that you saw in the other photographs that we were using on the tables. We would put the corals on the cookies. So we started doing that and then outplanting those. But when you're moving around in the boat, they would knock up into each other, and it didn't work out so well. So we said, you know what, we already been kind of doing microfragmenting directly in cement, because every time we have broken little pieces, we still put them in there. So that's what we're doing now. And when we cut the coral, the exposed bottom underneath is dead skeleton. So that's actually what's going into the cement, is the dead skeleton, not the live tissue. We've also evolved from having to always be on a key where there was a power source. And last year, the moorings have been donated a boat every year for spawning, and we got one for microfragmenting. So we were actually able to run both of the saws on the catamaran, pick up the corals, cut the corals, put them right on the reef. And like I said, this is amazing for us because it's just speeding up this whole process of how we can get to restore these shallow reefs really quickly. So last year, again, we do this course every January, and last year, while we demonstrated the microfragmenting, we didn't let anybody do it, but this year, all of you guys in our course are going to actually get to do it, because we have Dave here, so we can have the two saws going, and everybody's going to get a chance to do it. Last year, we just showed them. So for example, at Laughing Bird, this was my old way of doing things. Those are outplanted corals. They would have been in the nursery for a year or two before, maybe a year change, before I put them on the reef. Instead, we cut out these tiny bits and put them on the reef, and in a few, less than a year, they're growing together and making a coral, and I didn't have to put them in the nursery at all. Someone asked about other species. This is the pillar coral. While this is in the nursery, the pillar coral is that one really, also should be on the endangered species list. For Belize, we're still doing well with it. In Florida, they can count all 80, 90 colonies left. They've been ravaged by disease. They're reporting this coral having these terrible diseases in the Eastern Caribbean as well. I just got somebody on WhatsApp just sent me another photograph of a sick, dying pillar coral in the Eastern Caribbean. We're still okay in Belize so far. What I'm showing you here is that the lesion, that white bit, the dead skeleton, right? That's from the saw. And at day 90, it's already starting to grow over. And these are not like the acropores. They're slower growing. So anecdotally, Dave is saying to me that they're growing so much faster out in the sea, right, than they do in the controlled land-based areas, which is logical, right? Because they want to be out in the reef area. So these are just a few more examples of some of the issues now we have about talking about this stuff is how do we track it? If we put out a bunch of little pieces and they turn into one, no longer can we say we put out 300 corals and we end up with 30. That doesn't even make sense, right? Because then when they merge together, you have this nice little colony. So these are just some more examples. Those were at Whip Creek. This is at Blackbird and Turnef. And again, we're experimenting with putting several together or just one little one. And you can see the several together merge into one quite quickly and nice. And obviously it's the same genet. This is right at Laughing Bird Key. I was hoping to show you the surface because again, we're really concentrating in the shallow areas. Why? It's the shallow reefs that really give us the most shoreline protection, and that's where these species thrive. So hopefully you don't need the red circles anymore. Maya left me, there's no way to brighten the screen. It looks dark to me. But anyway, hopefully you can see that the little elkhorns here are doing quite fine in less than a year, and they're thriving and growing on this really shallow reef on the leeward side at Laughing Bird. We also started with annularis, and while the annularis, which is a star coral, is a super slow grower, you don't get those exciting time series. The point of this slide is they're not dead, so we were happy it was an experiment because one of the problems is most of the restoration has been focusing on the aquifers because they grow so fast. So if we can start using these methods to include all of our other important reef builders that typically grow much slower, and I'm pretty sure Dave's going to talk about that, it's going to step up the pace of what we're doing and the opportunities of what we can do for restoration. So in summary, on the microfragmenting parts, we are doing a little bit larger size classes for our work in situ, means in field, not on shore. Every time we do it, we want to do direct outplanting, but if the weather turns bad, and this is what we'll do this week, if we can't outplant, we'll put them in the nurseries. But ideally, our plan moving forward is just direct outplanting and have that backup plan every time if the wind shifts and we can't do the cement work. One caveat quickly for the course participants, when working with the microfragments, it actually gives us a lot more flexibility in the conditions we can work in. Why? Because if we have these giant staghorns that are big like this or elkhorns and you have that surge out there, we can't do cement work. It has to be flat, calm, because otherwise that thing is going to move around. And now that we're working with these small little flat pieces, we can get away with conditions to outplant in that we were unable to get away with before. So just pausing really quickly here if there's any questions on that, but really Dave is the expert on microfragmenting, and I show this photograph for the course participants because you see that little blenny there is like the size of my fingernail. You see the cement there, and one of the things you all should have a manual. We'll be going through the manual. We'll be with each other Tuesday, Wednesday, Thursday, that we emphasize every time out there, don't put cement on anything living. We're out there to put things back, not to harm anything else. And sometimes it's hard to wrap your mind around how small some of these things can be, but if we take our time, there's a lot of dead reef area where we're not going to harm something else. And the other thing that's hard to visualize, especially with the aquifers, is how quickly they can grow up, right? So we'll be talking about that later, about where we choose to put the corals, trying to think 5 years from now, you know, that it's not going to interfere with this star coral over here or that. That's a little tricky one. So I mentioned the genetics already. This is just illustrating what I meant. So if you can't afford, like we've been budgeting to send out to a genetics lab to get your genetics done, this is what I was describing. On the left, we have two different genets or individuals of the same species, and we know this because there's a seam when they touch, and one grows over the other. And on the right, we have two pieces of the same genet. It's the same individual, and we know this because when they touch, they grow together in a seam. So with these aquifers, we're fortunate. Some of you know about plants, like some philodendrons you can pick a cutting from, and that will root on its own. That's the same individual. So that's what we do in the nursery. We take cuttings of the same individuals and make more of them. But what I was mentioning and the importance of having the multiple individuals is for the sexual reproduction. We have to make sure that they can cross and make more baby corals out there. I thought I was done with the microfragmenting, but there's some time series up at Turnef, and this is showing you the point I'm talking about. You can see with your naked eye, there's two different types of elkhorn. Sometimes it's not obvious, but sometimes you can tell by the color and the shape that you have different genets. So like I said, we're experimenting with how close, how near we want them to be, and we've already worked with Dr. Bonds, the geneticist, on assessing our natural large sands and finding out how many individuals on the reef. I hope that's clear because it's kind of crucial. And this is an example where it's the same individual growing into each other. Okay, I'm not going to dwell. Don't worry about the graphs. I know you can't see them very well. The point of these graphs, this is switching to staghorn of the pipe shank. The point is simply that we've known for years all the individuals have different growth rates. As I mentioned before, they might have different bleaching patterns, different growth rates, different survival rates, and that is what we're tracking over time. Again, looking for the more thermally tolerant, the corals that are stronger, that can resist bleaching or bounce back from bleaching, and also disease and or maybe predation. What you can't see at the top is that since we've moved to saltwater key and Turneth, we found out that the average growth rates in general, everything is growing fastest down here in Placencia in the near shore environments versus middle and north part of Belize. So we have both a north to south gradient and a slight east to west gradient. And it's not temperature, I looked at that correlation, it's not depth, it's all the same depth. If we want to look at salinity, we're not sure. You can't see the numbers, but it's literally like 3 centimeters a month at the north, twice as much, 6 centimeters a month in the middle, and over 12 centimeters a month down here near Placencia. So it's a very striking difference. And again, these are average across multiple staghorn genets. It's not single genets I'm talking about. I don't have the answers for that yet, but we're going to work on that one soon. And there's nothing wrong with it, right? It just means that things are taking a little longer in Turneth than they are down here. So again, I was talking about how do we talk about this, how do we measure it, how do we share with our funders, with our stakeholders, with the tourists, what kind of results we're getting. So what we've been doing since 2014 is using photomosaics developed by Dr. Art Gleason. And earlier I mentioned now that we're microfragmenting, we might put out a bunch and they fuse together and it's less. But in other times, you might put out a few and then they grow and then they break and they catch and they make more. So how do we talk about survivorship? And not even just in Belize, as a restoration community across the Caribbean, we have not figured this one out, right? So the example that's easiest that I like to explain is prior to Hurricane Earl, which was a minor Category 1 indirect hit here in 2006, our very first 19 elkhorn corals we moved to Laughingburg, 17 of them lived, they're still out there today. You can do that math, right? 17 divided by 19, you have almost 90% survival rate. But before Hurricane Earl, I could count all of the little pieces that came from those original 17 corals, and there was 48. So how do you talk about that? That we went from 17 corals to 48 corals. I hope you're getting a sense of the frustration of how we discuss our results, survivorship, what we're getting out there, and that's why we have switched to the photomosaics. I have a few examples of what I mean. There are fancy softwares you can use. So we had a colleague at University of North Carolina use this thing called ImageJ. So, we put this as day 1 on the reef, and it was doing fine up to 2016. Then Hurricane Earl came and it flipped over. But guess what? It flipped over, it's stable, and it's growing back faster than ever before. So yeah, there's some fancy software. Somebody's grad student can look at all of that. But that's not appropriate for us to really be able to share and talk about the results we're getting. Here's another example. Day 1, when we put it out, a little nursery-grown elkhorn, Earl took its top off. But you can see it's already growing onto the reef. And then 4 years later, it's this thriving, beautiful colony, right? So again, how do we share this information? I think I have a few more like that. So you can 4 years later, and there are these giant, beautiful corals, the staghorn there. While we didn't put them directly there, they came from us, but storms and other things move them around, and they catch and they grow and they spread. Again, what is the vocabulary we use? How do we share this with people? We've moved on to the ropes. We grow them on the ropes, and to be even faster about getting them on the reef, some of the heavier ones, we just take the entire rope and set it down without even using any nails. This is out at Silks Keys. How do I talk about this? This is the growth in less than 2 years. It's thriving, it's catching. Again, what are the tools that we can share this information with? So what we've done is we've been using these photomosaics that Dr. Art Gleason developed, and many others are using now with this array of different cameras. It takes a zillion time interval photographs. You have a measured clock, and you're doing the zigzag pattern over it, and you also have video. And what it gives you, and we have 6 of these plots we've been tracking at Laughingburg since 2014, so it's actually 5 years of data now. And what it gives you is a composite image that then use another software to calculate the change in live coral cover. And so far, this seems to be our best tool to really talk about the results. I want to emphasize just one thing here. Look at this site 9. Can you see the pointer? Only 209 pieces of coral went out in 2010. This site right here. And we just got the results from 2018. And 36% of 39% live coral cover is from those original 209 fragments 8 years previous. I hope that's like sinking in. That's a big deal. That's like super exciting. What we really want to figure out is the minimum amount that we have to reseed in an area, and then we can move on to other areas, right? And let nature take its course. And again, if you pick the right sites to put them out, and you pick the right corals to propagate, and we have a lot of luck in Belize because we have some well-enforced no-take zones, and we still have some relatively good water quality. But that doesn't even really tell you what's going on at Laughing Bird Key, because we now have over 33 sites out there. And I've tried using drones, and I have no way to really share. You guys on the course tomorrow, and some of you have already been round and around that key, how do you really express how much coral is out there? It's mind-boggling. I think I have one last example. This tiny picture in the top was 2 sets of ropes with little pieces like this, 40 pieces of coral. They've been harvested, replanted with cement. We used nails, set down the 2 ropes. In 2012, you can still see sort of the shape of the ropes. And in 2018, it's like larger than this stage. I can't even fit it in a photograph anymore, and it's not one of our mosaic plots, right? So this is one of our difficulties. How do we really get this across? And this is an example of what I mean by photomosaics. And again, without even doing the high-tech annotation, which means telling the software what's in that image, you can see from the red circle to give you reference, all that other orange stuff right there is aquaphor corals, and we didn't add anything to them. So we didn't add anything to them since 2010, and they're just propagating, growing, asexually reproducing, sexually reproducing now too that we know of. And this is what it looks like in a graph. So the take-home message of the graph here, I know it's a little busy, but at least I made it rainbow colored for my hometown, San Francisco, right? But on the left-hand side, 24 and 23 were the only sites that were not outplanted. And the red bars are 2014. So they're really low numbers, less than 5%. That's because that's what that kind of coral cover was at Laughing Bird before we began restoration. We did not plant on those 2 sites. 20, 21, 9, and 13 on the right-hand side, we had already outplanted when we did that first mosaic. So you see a slightly higher number. But the take-home message here, again, we didn't add anything to them. In just 1 year, from 2014 to 2015, look how high that percentage of live coral cover jumped. And this is what I'm trying to get across, is it's so difficult to get this out there and to really talk about it and share it. And we're really hoping that as a restoration community in Belize, as people start moving on to do it in other areas, and certainly the Caribbean and even internationally, that we would start using these methods and make it more affordable and user-friendly, so we have a standardized way to talk about what we're doing, right? Because, as I mentioned, our vocabulary is lacking still. And look at this jump again. The drop in the yellow, I mentioned for Hurricane Earl in August 2016, indirect Category 1 hit. We mosaic like a week after that. So it flattened some of our sites. But look at the jump from the yellow to the blue. It seems like a slight disturbance, if it's not catastrophic, is actually regenerating even higher and faster growth of these corals than the initial growth. So we're really excited about that. We are looking at sites now with over 40% live coral cover. The average in Belize right now is 16%. So this is phenomenal results. We're really thrilled. I'll skip that detailed slide. This is representing the graph that most people like to see better than the graph because it's an actual time series. Little baby corals were less than a year old on the left, and then that photograph is the exact same site and coral from 2016. So for most people that is more meaningful than the graph. Hopefully the graph made sense. But now, because we're going more and more shallow, we have some challenges with the mosaics because, I don't have the right terms, but because of the way the photograph needs to be taken, if you don't have enough space between the way the photos are stitched together and everything like that. So we wanted to be working with drones. We haven't gotten the results we wanted out of the drones as yet, so we're still working on that with Dr. Art Leeson. This I'm not going to dwell on. I just want to let you know that all those mosaics information I told you about, we can get a lot more than just live coral cover on there. We can look at sponges, we can look at crustoscoralline algae for my course participants. We can look at all the benthic organisms. And so we can get really more information about what's changing on the reef now that we're adding all these acroporids. And for fish people, we are doing fish. This is the first year set of data. We now have that data set through 2018, but I tend to give all of that fish data to Boston University consultants and my friends because fish just move around too much for me. So we have to get a lot of data and a lot of statistics, and I'm working and waiting with Boston University to put together the results that we have, but we are looking at the fish on all of our sites since 2014. Even more important than the fish to me is all of the things that skip regular protocols. So for example, in AGRA or in MVRS, which are mandated protocols that are done in all of our NPAs and protected areas, you're looking at primarily commercial species and maybe some herbivorous species like parrotfish, but you're not capturing everything. You're not looking at blennies, you're not looking at hamlets, you're not looking at triple fin. I mean, there's just all these different little critters out there that use these reefs, and what we really wanted to capture is the biodiversity that comes along with what's known as foundation species, the acroporids. Foundation or keystone is like the forest and the trees in the forest. You have something there that all these other things live on or amongst or whatever, and this is what we're trying to show, because one of the other criticisms that we still hear all the time is you can't restore a reef with only a few species. But what we're trying to show is that actually, yes, these species bring in a lot more species. So my favorite part, and Jason's going to talk about it some more, is I've been showing this picture for years now about all the different crabs that are true to our nurseries. So when I met Jason and he had all this data to show how much algae the crabs eat, I was like, oh my God. So finally, 4 years later, 5 years later, actually almost 10 years since we first thought about all of this, we now have Jason and Mark Butler here, and they're also working with Dave in Florida with the crabs and the corals. And a few points I want to make here, and that is that this one, the middle row, middle picture, this one apparently can fetch, I've been told, for as high as $6 to $12 US in the aquarium trade because it cleans all the tanks, right? That's how much algae it eats. So we can start to wrap our minds around, if we're already growing corals, what about sustainable mariculture for the aquarium trade? That's not right now, that's not today, but that's something to think about. But what's closer is this bottom one here, the Spinosystemis, and I can't pronounce this genus name, but it's the big one that we like to eat, the double crab, right? So this guy eats all kind of algae. We see them all the time. I have 10 years of photographs and videos in the acroporids. I asked Mark, like, isn't there a correlation with these crabs and acroporids? And Mark, if you remember, he said, well, there's no acroporids in Florida, so I don't know, right? So sorry, Florida. But, so I'm glad they're all here. And then the last slide I want to show you here is this, I don't know if you can see it, there's actually a crab claw sticking out of the hamlet mouth, the mouth of the fish. So the staghorn, these porcelain crabs and all these other crabs live there, and then here comes the fish and they're eating the crabs, they're getting habitat, they're getting food. And my point of that is that we are actually restoring an entire food web ecosystem with just that staghorn, even though that was such a big criticism in the beginning, right? I think that I have a video I wanted to show you that's not included. The idea we have for the crab is I need to rename this and not really call it farming but restocking, because the idea that we have based on a lot of people's work in previous decades is that the females, and I think I'll let Jason talk about most of this, but it turns out they're really easy to grow. And we already have fishermen here that have these cement lobster shades in our area, over 100 of them. They've been on the reef for 10 years. You can see they look like the reef. When we went out with Dale to find some of his shades, we couldn't even find them. We had to say, sorry Dale, you gotta get the water because we can't even find your shades, because that's how much they look like the reef, right? And so besides lobster, the crabs themselves recruit to these shades. So we want to work with what's already out there. I wanted to show you, can I just escape like this? Show a quick video. Will it bring me back to the same place? Because my course participants, we cannot go to French Louis Quay. I hope this works. And I don't want any volume. Is it turned off? This, some of you guys may have seen this out here before. This was actually at French Louis Key, so Mr. Ronnie, rest in peace, sold the key. And they pen off these shallow areas in the mangroves. This is what I wanted to show you guys. And they would keep the lobsters in there so that when they have a guest, it's fresh and they're already there. And conch. And this is the idea that we're hoping we can sort of twist and use for the crabs. So if we could bring in the buried females and figure out a way to capitalize on the research that's already been done so that when they let go their eggs, they have a place to land, and then figure out a way how we feed them, how big they need to be. We're actually not farming them per se because that female can go right back out on the reef because they breed year round. So when she does go to eggs, she doesn't die, she goes back on the reef. And then if we can figure out the size that we need to get them to, then we could simply put them back in all the guys' lobster shakes. And I guess restocking is the best word, or maybe you can come up with a better word, but it's sort of enhancing that survivorship of those juvenile crabs that normally everything in the world eats, right? So I don't know if you remember the stats on, like they say with a turtle, like there's 1 in 100,000 or whatever that, survive to sexual maturity, and you probably know the numbers of the whales are even worse, right? So this is, we can't go to this key, we will go to a different key tomorrow, but so I wanted to show you guys what I'm talking about, how we could work with some of our inner keys here, low infrastructure, all in situ, hopefully get fishers involved and things like that. I'll stop it because this is 6 minutes long and I need to wrap this up. But you guys get the sense of that, right? Yeah. Okay, so that was crabs. Success. I just really want to quickly mention, because I'm running out of time, that I mentioned all that thermal tolerance stuff to you too. I didn't go into basic coral biology because I'm sorry, all of my course participants should know this already. They know what bleaching is, but we also do bleaching monitoring. We had a very severe event in 2017, which we had not seen for a long time, so we have a lot of that data. It's stuff that we're looking at, trends north to south. We're matching it with temperature, long-term, and recent temperature data. And again, we need to get back on track on what we call resilience and thermal tolerance. So I'm not going to dwell on it. I just want to let you know that we're looking at all of that. For example, why some corals might bleach completely and recover within a month or two, why some don't bleach at all, and why some bleach and die. And so if we can figure this out, again, we'll be a long ways forward. I'm not going to dwell on all of this, but again, we have temperature data all over the place for many years. This is a borrowed slide from a partner from Healthy Reefs Initiative. And this is simply showing that, yeah, we know 98 was terrible, 2010, 2015, but this clearly shows that 2017 was worse than all of those years for Mesoamerica. It's not always the same pattern for the Eastern Caribbean and Florida as it is for Mesoamerica. And even within Belize, it can be different in north to south. So this is 2017. These are nursery-grown, outplanted corals at Laufenberg Key, super shallow, 1 to 2 meters. They didn't even bleach. They were not bothered at all. And yet at Saltwater Key, in a channel with high flowing water and a little bit deeper, all these other species are fully bleached. So this is what I am terming resilience, right? So that is really a resistance rather. That's resistance. I mean, they didn't even bleach. So this is what we're looking for and aiming for. I just have a few other photographs of what is resilience then, right? So these corals bleached, but then they recovered. They did not die. This is out at Gladden Spit, all the way out at the far outer reef. Again, it bleached in 2017, did not die, recovered in 2018. There's a photograph of the person in there, a size, an annularis coral colony this size. Dave, want to care to guess how old a colony like this is? This is like, I mean, these are hundreds and hundreds of years old, these corals. So we really want to try to understand why some of them are, making it and why others don't, and of course propagate the ones that can make it. I mentioned already we've documented the spawning. I'm speeding up because I want to let all my other speakers get to talk. We have future plans of doing working within the MAR Fund, Mesoamerican, which is Mexico, Guatemala, Honduras, and Belize. And we're starting to move forward on how we can work with each other. We did an exchange already, so that if there's a bad event, like not a brownie, but like a hurricane, and for example, example, when Iris came here, we couldn't worry about the beef, we were worried about homes and families and things like this, but if we had people trained that were not unaffected, in theory they would come down and help us with the beef, while the people that live in that site are trying to get their homes back together. And so it's the same idea, like reciprocal action. If something happened in the Yucatan and we're already set up and ready to move, those people in the Yucatan may be scrambling to get their lives in order, whereas a trained team could go out to their reef sites and in theory save back some of those reef sites before they're ruined. And then the photo mosaics I mentioned, we're looking for funding to do a workshop just on that because we have a lot of feedback from people that want to learn just that. The microfragmenting, some of you will get this week. And of course, I mentioned this actual course is an annual course we've been doing since 2015. I hope this video plays and then I'm almost done. So these are just some fun, oh, I didn't link this video. Well, so the video is going to show that we have sea hares. It was the walking crab all over the staghorn and in my video. And so the point is, it's not just one or two things out there, right? These ecosystems are fairly new to us. We've only been going underwater really since the 50s and 60s, and we're still learning a lot about what all these relationships are. And we have to look at it as a holistic picture. And with that, I'm going to emphasize that in Belize, we've had many consultations over the years, and unlike other parts of the Caribbean, we have decided as a group that we are training coastal community members, meaning tour guides, fishers, NGO staff. They get trained, they learn how to do this work, and then they get paid equivalent to a dive master stipend. Because not only because Belizeans need jobs, that's not the main point, that's a point, but the other point is that when you have the same people doing the work over and over again, those people become more efficient, they know what they're doing. So today, I can take out a team of 4 to 6 people and we could plant 2,000 to 4,000 corals, but if you give me 30 outside volunteers, no matter what their good intentions are, I can't do anything with them, you know what I mean? So there's a lot of reasons why we want to emphasize keeping to do these training, sharing these knowledge methods and learning as we go along. And even though people come and go, if we can keep this going, maybe, somebody busy tomorrow, but that person suddenly free up that took the course last year, this kind of thing, the more we do it, the more we can have some standardization of the way that we've been doing things and hopefully keep getting the good results we've been getting. I hope that's clear. I'm going to skip through all of our collaborators because you guys probably don't know them anyway. But we did get a little accolade from the Village Council. We were thrilled about just last year. Fragments of Hope is very small, so we just have our first full-time outreach officer, Monique Vernon, just came on board with us last May, and that is through private sector donation. And so everyone else, all the other green, are the over 40 people that have worked with us in the field at sea, boat captains, fishers, guides. We have boards, but those boards are not paid, they're volunteers. So we're trying to figure out a way where we could get a few more full and part-time people involved so that we can get even more work done and start to spread this out and ease up my workload. And then we had a little bit of Brata. We got some awards. We went to COP23. We have the exchanges I have to mention with Mexico last year and this year under MAR Fund. There's a picture of Mr. Mahil. He passed away a few years ago and he was a huge supporter. He and I got to go to Japan. Japs and Hut, who also recently passed away through a seaweed program, got to go to Colombia because of our original exchange with corals in Colombia. I have Wilbur Dubon in there that also worked with the seaweed group and got to go to GCFI under one of the Fragments of Hope grants through GEF. And that is Valentine planting some mangroves, so we've also done some mangrove replanting work, mostly with GEF small grants funds as well. And we're working, and we have partners in Colombia, so they've adopted the dome. That picture is actually not from San Andreas. The dome is from Providence, which is the next island over, and they just gave me that. St. Bart's in the Eastern Caribbean came here as well. Now they're using these domes for the staghorn, and they were using the tables for a while as well. And just for those of you that don't know us, you can go to our website, you can find this information. But we wrote our mandates so that even though we mostly focus on coral restoration, it's not just that. We also have capacity building, outreach and education, a lot of things going on. And in summary, so I can let all my other experts speak. These are the stats. So we are now in over 10 different areas. Not every nursery site is pictured. The circles by False and Mohoki mean that those are outside of MPAs. They're not protected areas. So those are long-term controlled experiments so that we can really start to quantify and evaluate what we believe anecdotally is so important, is that no-take zone to protect that entire ecosystem. And the numbers, again, I hope you understand now how they're sort of meaningless, but the numbers that I'm showing you are when we outplanted almost 25,000 corals in 2018 alone. And then the summary numbers are for each of those sites in total. So there's over 80,000 corals allowed in Bird Key, but when you swim around the key, again, we'll see how that's kind of meaningless because it's just these fields and fields of corals. So with that, I'm going to finish up, and I'm actually, we're at 5 o'clock, so what I want to say is I'm going to have Dave and Mark and Jason introduce themselves again, and we are only here until 6 p.m. I apologize because there's a higher commitment for the room at 6:30, and we do have tamales and samosas in the back. Normally we wait till the end to eat, but maybe we ask people, like if you're starving or peckish, you just get one tamale and one samosa each so everybody can get some. But with that, because I want to move people, we only have an hour, so I want to get to Dave and Mark and Jason and all the good stuff right now. So I think I'll ask you to also hold questions until the very end so we can make sure we hear from Dave and Mark and Jason because they might need to cover that question that you were going to ask. And I'm sorry, but we are short. We only have one more hour. So with that, let me introduce Dr. David Vaughn again. And let's put your slideshow up. And for those of you that haven't seen or checked in yet, make sure we have this large these artwork we've been using Anton Leslie's, so we have the large original More Corals Equal More Fish posters up front. They're not for your living rooms, please. They're for public places like schools, dive shops, if you have a hotel, you know, some place where it can be shared in the public. You may have seen our billboards we have down at the dock, we have by the airstrip. Again, thanks in large part to GEF and some other funders. And we just installed a billboard at Laughing Bird Key and Moho Key just last week. And we have the second image poster, a smaller poster because we did it in country in the back, again from Anton Leslie. And somewhere I have an example, not to give away, the posters can go, but the puzzles are for the schools. And so we also did coloring books. We have an example of the coloring book in the back. The coloring books can be downloaded from our website as a PDF. And these coloring books, again, are the inside is each of those individuals in these composite paintings. And then the jigsaws for the schools as well. So we have a lot going on. Make sure, again, the puzzles are not to give away, but the posters and the coloring books, you guys can all get one of those. Now it's Dave. Yay! Thanks for having me, Dave. Did you want to mic up Dave too? Can we just transfer this?
That'd be great.
You know what you're doing, right? Oh. Doing your own arrows, or you want me to stand here and do it?
I don't need that. Just the floor.
Yeah, this one. I think so.
Well, hello everybody. I want you to give it up for Lisa. Because she's taken over a decade or more of work and scrunched it into 45 minutes. I don't know how she does that. But as you can see, she can talk very fast in order to try and do that. I'm not going to try to talk so fast, but I want you all to know how special Fragments of Hope and Lisa and the whole group that has made this possible are. I've tried to pick out a half a dozen places around the world that have really made a difference in corals, and this is one of them. You're living in the area that actually has done one of the most incredible jobs of putting together the ability to restore corals at a scale that you can see. You don't even need the graphs when you see the pictures. When you get to see it in person, it's just amazing. And the problem we have is it's just that it's a big world. And so really the question is, how do we get this to scale? This is a great start. And we've done this, a number of us around the world, really feeling like we're people with a shovel and an axe. And in the future, as you see some of the new technology from photomosaics and genetics and microfragmentation and the rest, we're actually able now, hopefully, to use technologies to bring us into the inner space stage instead of what happened with us having our moonshot from never being able to send a rocket up in just a few decades. And now it's our turn in the inner space to make the next moonshot work. But how do we do that? How do you do that with scale? So I'm going to try to take you through a few of the steps of what I think is some ideas, and a lot of them were tested and discovered here as well, and a few other places, of what I think the next steps will be. So I worked for about a decade and a half at Marine Laboratories in the Florida Keys. I like to say I retired 2 weeks ago, but I like to say I graduated 2 weeks ago. And if anybody has seen my AARP video, yeah, that stands for American Association of Retired People. It says that I'm not going to really retire until I plant 1 million corals. So the name of my new company is Plant 1 Million Corals, the new shirt, and hopefully this will be one of many places that show that those kinds of things are possible, because planting 1 million corals is actually just a good start. We actually hope to change the name of this process to Plant 1 Billion Corals, because that's what it's going to take, like it did with planting 1 million trees, which became, for the Nature Conservancy, Plant 1 Billion trees in the rainforest, which is now somewhere between 7 and 8 billion trees that have been planted back in the rainforest. So those kinds of things are possible to do. But I'm going to take you back actually into aquaculture. I actually did aquaculture of many marine species for about 30 years first. And I'm going to take you back to really aquaculture 101. In marine species, really, all marine species, aquaculture is made up of a hatchery, a nursery, and a grow-out or outplanting. And the nursery could be either a land or a field or both. And basically, just like whether you're growing shrimp or fish or oysters or crabs or corals, the hatchery has controlled conditions indoors on land. So does the land nursery, has a little bit more. The field nursery really relies on faster growth because of natural sunlight, natural conditions, and the outplanting is where the animals really take off. So you want to push things as fast as you can to the outer area, as long as they can survive. But if you see this circle, this is where corals started. Corals started only with the outplanting and only with the field nursery, and mostly because those two acroporid species, okay, it's the branching corals, the staghorn, the elkhorn that looks like a moon's antlers. Those two species actually evolved as small storms if they broke and they landed in a hard spot and stayed sitting in one spot for a while, didn't roll around in the mud or the sand, they could actually grow and attach themselves back. And they actually utilized the asexual reproduction of fragmenting, breaking, and that made it very easy to, in the field, a field nursery, just by hand or a pair of clippers, cutting corals into a 1-inch piece, and in a year it was 10 inches and ready to plant. So it was a very easy way to get started. And by the way, people didn't even look at the typical way everybody else does aquaculture. Everybody else does aquaculture. If you're familiar with the shrimp hatcheries that were here, they start with a hatchery or a fish hatchery. They actually spawn, clams or oysters or fish or shrimp. They take the millions of eggs that are out there and they try to get many more of them in controlled conditions to survive. Well, in corals, that has not been the case. We actually didn't know that corals had a sexual reproduction cycle until the 1980s. Could you imagine us today saying, oh, we don't know how horses breed. We're just not sure. There seems to be more of them now and then. And that's what we found out once in Australia in their summer of the year, near the full moon, exactly 2 days after the full moon, exactly 2 days after 2 hours after sunset, in the middle of the night, boom, they release all these sperms and eggs in bundles, and maybe 1 in a million makes it. Every 25 to 100 years. Now that sounds crazy, but a lot of these corals, like we saw Lisa show the picture of one, that was probably a 500-year-old coral head. And so it only has to be successful once 100 years to produce 5 if it lives 500 years old. So everybody worked with what we call fragmentation, and as Lisa mentioned, by mistake one time, about 15 years ago, I said, I'm used to growing corals, plants and oysters and fish with a hatchery. I'm not used to taking a fish, breaking it in half and making 2 fish. I said, there's only one person I think did that. I'm not stepping into his shoes. I'm glad you got it. And so I said, let's try the sexual cycle. And I knew that 1 million eggs and only maybe 1 in a million make it, but only people never had it. The good news is we had 1 million eggs of Elkhorn coral, and we made the first 11 test tube baby corals about 14 years ago. However, if I told you I only got 11 to make it at 1 million, and not told you that 1 in a million makes it every 100 years, you would have thought, no, that's not too much to brag about. But at 1 year old, that Elkhorn baby coral was only the size of a pencil eraser. 2 years old, the size of a coin. 3 years old, the size of a large coin. And I got disgusted with it, put it on the bottom of the aquarium tank and forgot about it for half a year, went to move it, and it did what any good branching coral does. It grew onto the bottom of the coral tank. And I didn't know it. I went to move it. It stuck, so I yanked it. I heard a crack. It broke it into tiny pieces. And I thought all the tiny pieces were going to die, and that's not what happened. They all grew 25 to 40 times faster. And they all grew back up to the size of a 3-year-old in literally weeks or months. So I quickly, like a scientist, grabbed a scalpel and chopped them all up again, and it did the same thing all over again. And we tried it with all 28 species of corals. Mostly the big massive corals, and it works with all of them. So it's a new deal for what we call microfragmentation on land. But I want to show you some new things too that have taken place here to show that we can actually do some of that fragmentation at sea, like you saw pictures of, and quickly put them not only back in a land nursery, not only even back in a field nursery, but really right back to the field out plant. So these are game changers that will make things to scale actually possible. And a lot of them are taking place here. We used to take a big tile saw, take something about the size of a golf ball, cut it into pieces, and wait years for it to grow back to the same size. We now in our land-based nursery have this diving saw that's made for making coral jewelry, little tiny pieces like the size of earrings. And we cut these corals on land into these small, tiny pieces. Not only does it give us more, in fact, in one day we can cut 1,000 pieces. We used to cut a couple, the old-fashioned way, we took 6 years to produce our first 600 corals. And now we can produce 1,000 in a day, and in 6 to 12 months they're up to size and ready to go. So it's a game changer for corals, and it's nice. And it's a game changer not only for just making the coral more corals, but if you see here, this is the large fragment size, and this is the number of years across the bottom, and what an increase. This is how you get to scale. And so now we have produced over 60,000 corals, 25,000 planted just this past year. And so it makes it to some sort of scale that you start to see a difference at a reef lake. So the microfragments, we used to take a piece the size of a golf ball and cut it into two, wait 2 to 3 years, and we do it again. Now we take that same piece, and if it's a palmata, the big elk coral, we'll cut it into 100 tiny microfrags, or if it's one of the bigger brain corals, we might cut it into 20 to 50 pieces. So we get a lot of pieces in one time. Not only that, we start making more corals than we had space for them to grow. In the past, if we cut some corals, we'd have 2 years before they grew up and we'd have to do something with it. We are now cutting corals faster than we can buy and get manufactured new tanks to hold them. And corals, you usually don't want to hold a coral close to each other because 2 different corals will try to fight. And we couldn't afford to do that, so we put the corals close together because we had to cram so many new coral fragments into one tank. If you look on the right, those are the ones on the left. It's a square, it's a 1-inch square. It has a little piece about the size of a pencil eraser on it. And then in just about 6 months, you see them on the right, they're about the size of a fat golf ball. And you might want to see something. On the right-hand side, you see kind of a mixture of blurring, because those corals are not only touching each other, they're not fighting. And they're not fighting because they all came from the same parent. So from one which made 100, if we put all 100 near each other, they touch each other, just like your skin being transplanted to your other arm, your skin notices that it's you and it doesn't reject it. The corals aren't rejecting each other if it came from the same clone parent. And so they're actually fusing back together. And so this gives us a new tool that we can actually scale up not only a number. For the first 6 years, we had 1 raceway. Now we have 70 raceways, like you see on the picture of the right. That's one way to scale. But you can also do it in just because of the fixed cost of the prices. These are 3 different entities. One is the Staghorn Project, one is the Oceans, Reefs, and Aquariums, a marine ornamental company that I ran for a number of years with Robert Ranchers and McGrath. And what is our massive corals? And what you see is they started off at $100 to $250 a coral to hold it for 2 years in a tank, and now we're down near $10 to $20 a coral, and I predict in just the next few years will be to $1 to $2 a coral. This is how you get the scale. It's not only the production in numbers, but it's the production in lowering the cost. And really what you see here is this is the coral numbers at the top, going from left to right and increasing. This is the cost going down. That's because of your fixed costs are there, but you're producing now 10 times more each year at the same cost. Your costs come down. So this is how to scale in number, and this is how to scale in lowering the cost. So I don't know if anybody remember the old film 5 Easy Pieces, but I figured 5 easy pieces to help, with goals. First of all, we got to take what people always ask me, why are you making a lot more corals when we know that worldwide the corals are dying? What we're doing is we're taking what Lisa described as the resistant or resilient species, strains, types that made it today, and we're making more of those. So that we need to make sure there's not 10% coral cover out there, but 50% coral cover or better. And so we're also, in a hatchery, if you were making new corals by combining new eggs and sperm, you're making new genetic strains. So that's one of the things that will happen in the future. The other thing is it's something that started in Florida and really showed itself here and has also showed itself in Cancun in Mexico. Microfragmentation not only can be done on land, but it can be done in the field nursery, and maybe directly out, and maybe even underwater. So we've seen here with the first time, and I have to tell you this story when I show it, is that I showed up here, and Lisa wanted me to bring that little saw, and I brought that saw, and when I got here, I said, where's the land-based tank that I'm gonna, do all this cutting with? She goes, get your ass on the boat. I said, what? She said, get on board. We're taking that with us. So we put that in a generator. We went out to the field, and we started cutting on a dock with a generator. And the first day we planted some and put them right into the field nursery and they lived. The next day we cut some more, and then the wind blew up and we couldn't get to the field nursery. She goes, we don't have a chance. We're just going to plant them right out. And we did. And a few months later, she showed me pictures. And I think microfragmentation in the field was born here. And I think that's going to be a very neat thing in the future. So we can do fragmentation in a field nursery. I'll show you some pictures of what we're doing. And I think we can actually do something new that we tried in Mexico this year, is we actually took our first plants and we did microfragmentation of the field plant underwater right to the bottom. And it's working as well. So in the land-based hatchery, it really is not rocket science. I call it bucket science. We actually take the spawn, collect it from the wild, eggs and sperm bundles, bring it into the laboratory, and we actually work it in buckets. So it's an easy system. You can see the picture at the top of the bucket on the right, where you can see individual coral larvae, and then we settle them. And they are very tiny, and it's amazing that these things actually make it in the wild. At 1 to 3 months old, you still have to take a picture under a microscope so you can see them. At a half a year old, you see them on the right. This is actually 1 millimeter, the size of the head of a pig at a half a year old. It now has the algal component in it, and most of these in the field would not make it. But all we need to do is get it to a year to 2 years to 3 years, so we have a new genotype, a new genetic combination of 2 different parents now that have come together. Hopefully parents that have natural resistance and resiliency to survive. And we did that early on in 2005, and we got our first 11. Then in 2013, we got 25. In 2014, we got 30. We now get 1 to 5,000 new genotypes every year. And that makes it so that we now have new ones. The new microfragments that we make from those genotypes actually grow pretty quick. This is a 5-inch diameter disc, and this is 4 little microfragments, and every one you move to the right until the very bottom row is actually fully fused is just 1 week. So this is 1 week, 11 weeks old, and you can see how fast they grow. We can do a hatchery and then make microfragments and do everything else out in the field. And so we can make microfragments with those. And how do we get to scale? I'm going to show you the math. For elkhorn corals, we get 50 to 100 pieces. So if we start with 400 elkhorn corals times 50, we cut into 50, we have 20,000. In 6 months, we're ready to cut again. 6 months, cutting again 20,000. There you go. 1 million, I can retire. All I need is 1 year. So here's the Fragenfly. We started this in Florida. In our field nursery, we weren't allowed to plant anymore, so I took the stem, you see it, and I just took clippers, and I went clip, 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 and produced about 200 fragments on the boat in about 20 minutes. And this is what you saw, the pictures Lisa already showed, that seeing is believing, direct outplants from clipping on board on a catamaran and putting it right back in. And this is what we've done in Mexico. This is taking an existing 5-year-old outplant, which you've got plenty. We take the same clippers, and we take a clipping in the field, and we put it immediately 1 meter or 1 arm's length away. And so for every one, we've gotten 10 to 20. And it looks like about every 2 months they heal up like this is pictured, and we can do it again. So if every time we plant one out there, we can. And so we plant now some of these identical pieces which fuse back together. This is a 800-year to 1,000-year-old coral head. It's about 15 feet across, 12 feet tall. We put 800 pieces on that. We're actually going to bring it back to life. So how do we go back into this? Well, everybody doesn't need to be a hatchery. Please don't do that. That's not what they do in shrimp. One hatchery supplies 10 nurseries, which supplies 10 grow-outs, etc. So if there's one hatchery in a region, and it supplies 10 field nurseries, it supplies 10 field nurseries, or land nurseries, 10 field nurseries is 100. And if they each have 10 planting groups of fishermen that actually plant out, then each group can plant 10,000 and you get to the million as well. So what do we need to do to plant at scale? A couple of things. We actually have to do production in the millions. We have to get the cost down to $1 or $2 per coral. We need permitting that does not restrict the first two. This is cases in other places. We need to monitor technologies at scale, microfragmentation, new genotypes, and outplant earlier and right in the field like it's being done here. Recut the ones we have planted a few years a couple of times. And please, let's start making and letting fishermen and divers plant corals, not scientists. So get started and keep moving, and it's a good first stage. Thank you. So I guess, Lisa, do you want to introduce?
Thank you, Dave. Next up, we have Jason Spadero. This is your man who needs to see USB drive. You're going to use this one, right? Sure. Right there. Wait, Mike. Sorry.
Yeah, so there's water at the back and also samosas and tamales.
We okay forwarding yourself? Okay. Sorry to keep moving, guys, but we have one more talk after this and time for questions. So like I said, I apologize, but they have yoga here every Monday, so they already asked them to start late for us. So Jason, maybe you could just introduce yourself better than I did. A couple sentences.
My name is Jason. I'm a PhD candidate at Old Dominion University. And I'm here to talk about some of my dissertation work that looked at using crabs to improve the success of what Lisa and Dave were just talking about. We'll get on to it. So Dave and Lisa both talked about putting corals back on the reef and restoring reefs and the giant effort around the globe, particularly in the Caribbean, that's being put into trying to get corals back on the reef. But why? Why the hell do we care? Coral reefs are busy places, right? They've got lots of stuff going on and lots of stuff that's there doing stuff that's only done there. And some of the stuff, or actually a lot of the stuff, is only found on coral reefs. So in other words, coral reefs only cover about 1/10% of the seafloor. But they contain something like 25 or 33% of the species in the sea. So they're incredibly important to biodiversity. They're also really economically valuable. Around the world, they generate about $375 billion in goods and services every year. So not only are they intrinsically valuable for biodiversity and all these different species that occur nowhere else, but they're also very, very economically valuable. Especially to places like Belize, on all these nations that rely so heavily on tourism and fishing and like activities. So it's all the more alarming. I apologize for the crudeness of this slide. It's all the more alarming with this high value that we haven't treated them so well. Yeah, for a long time we've been pulling a lot of fish and other stuff off of them. The oceans the pH of our oceans is dropping, and that's not good for stuff that uses calcium carbonate as its skeleton. And our seas are getting warmer, and all of these are contributing to, that just means that they're changing. We're going from, let's say, think of reefs on this graph, where on the vertical axis we have corals, and at the top we have lots of coral, and at the bottom we have not a lot of coral. And then on the bottom we have herbivores, stuff that's eating let you know, the stuff that's eating vegetables. You know, and on the your right, there's a lot of herbivores, and on the left, there's not a lot of herbivores. And so when we have lots of coral and lots of stuff eating green stuff, we have that healthy condition. So we have coral dominate the reef. And then on the bottom, when we don't have a lot of coral or we don't have a lot of herbivores, we get this nasty, degraded, unhealthy reef. And then this black band in the middle is an unstable equilibrium. So each of these two states, so when you have lots of coral and lots of herbivores, you're stable in coral dominant or healthy state. And when you don't have that, you're stable in a condition where you're degraded. And this black band that runs through the two is this unstable equilibrium state. I like to think of it as if you had a hill, a bell curve that's a hill, and you're trying to push a bowling ball up from one side. It's very stable where it is at the bottom. It takes a lot of force to get it up over. And then there's this one little spot at the top where it will sit on its own, but all it takes is just a little push and it'll go down to the other one. The same thing happens with these coral reefs, where if we have herbivores drop below a certain point, or corals drop below a certain point, or both, you set off these feedbacks where the reef flows in one direction or the other, just being reinforced by these negative feedback interactions. So back in prehistory, the reefs in the Caribbean looked just like that was a joke. Okay. The reefs in the Caribbean were in kind of this state. We had lots of coral, and we had lots of stuff eating stuff that wasn't coral. And then in 1983, we had a series, well, actually through the 80s and into the 90s, we had a lot of stuff that happened that pushed us, very, very hard back against this herbivore thing, or a factor. And then in the 90s, and the late 80s and 90s, we had several coral disease outbreaks that dropped corals quite a bit. So now we're currently kind of in this same area, especially with some of the diseases that are cropping up now. And what Dave and Lisa were just talking about is a whole concerted effort to move these reefs in the Caribbean back into this coral dominant stable state, or healthy state, by manipulating coral. So obviously if we put coral back on the reef, coral cover goes up. What we looked at was, there's a lot of people doing that. And here's kind of a representative photo of Ursula last month. Mark and I decided to look at the other direction, and not just can we push the reef across this axis until it gets into that state, but no, if you look at it, if we start at the base of this arrow and we move the reef across this way, it has a shorter distance to go with coral cover to get back across that line. And that's the basis for my dissertation right here. And it's not new. There's been a lot of legislation that says don't kill parrotfish because they eat algae. Lisa showed a video where they were nipping algae in one of the nurseries. But parrotfish move around. Their effect isn't concentrated in the area, so it might be very big, but it's dispersed across the entire reef. Diadema, the urchins, the long-spine nasty sea urchins that are out there, they were very effective grazers, and they were so abundant that they mowed down algae, but they also kill off those little baby corals that Dave was talking about. They just eat everything. And that nasty event that happened in 1983 that pushed us across here was all of these died throughout the entire region. And now, 35 years later, they still haven't made a meaningful recovery. So we said, why not, what else can we do? And Mark and I looked at this crab that apparently has left to eat down here. And you're right, the name has changed 3 times in 4 years. So it's different. This is Maglemithrax spinocismus. It's the Caribbean king crab. It's the largest spider crab in the Western Atlantic. And it is now named Maglemithrax for Tobey Maguire, who played Spider-Man. It's awful. Yeah. Taxonomists need to be taken back. So it's very large. This is one that's up in Florida that we pulled in a couple of of years ago. And these are actually small compared to the ones that I've been finding in Belize over the last couple of years. But what's more remarkable, all right, so more remarkable than its size is that it eats algae. And it eats a lot of algae. And it eats a lot of algae that other stuff won't eat. Algae are bad for coral because they compete for space and other resources. I'm missing a couple of slides. But we decided to test, basically, these crabs are out on our reefs naturally. We find them in Florida and Belize and throughout the Caribbean. But their density is so low that even though they eat lots of algae, there's so few of them that you don't see a measured effect. But what happens if we mess with the decimal point with density? If we aggregate them or increase their density or enhance their abundance on a reef, what happens? What effect do they have on the benthic algal community? And so we decided to use near-shore patch reefs in the Florida Keys to test this. And they're great because they're abundant, they're replicable, they're easily manipulated, beautiful experimental units. Oh, I don't even have that. All right, so I chose 12 of these in this first experiment. And on one set of 4, I broke them up into 3 groups. So one set of 4, I did nothing to. The next set of 4, I put crabs out on them at, so 1 crab per square meter of surface area. And then on a third set, I went out and physically removed all of the algae and then put the crabs out to see if they needed a little boost to maintain that. And then I came back once a month for a year and took several haphazardly placed photos of the bottom to track changes in the bottom through time. And this is a summary of those results. So on the vertical axis is the percent cover of macroalgae on the bottom. And then across the x-axis is the different sampling events. So the reefs that I didn't manipulate, algae cover was very high, and it stayed very high through the whole year. But just by putting crabs out, we had an 85% reduction in the cover of macroalgae, which is amazing. That's incredible. And then by scrubbing and then putting crabs out, we dropped the algae down and kept it that way, which is fantastic. But when you take this as a graduate student to your major advisor, he said, BS, do it again. So we did. We moved it about 10 miles away to another set of reefs in another year and redid the entire experiment, but added another group where I removed all the algae and didn't put crabs out just to see what happened. And this green bar is the control, where I didn't manipulate anything. I just came back and took pictures, this time 4 times throughout the year. And you can see that it was high. It wasn't quite as high, but it stayed that way. And then by just removing algae, we had all kinds of weird stuff. But by the end, the point is that it came back to almost what it was before we started. So unless we put the crabs out, algae come back. Just by putting crabs out again, we had about a 50% reduction in the cover of macroalgae. And then by removing the algae and putting crabs out, we had the same effect. So we think that we have demonstrated, or at least supported, the results of the first experiment here. And now we've replicated it twice, so we're pretty confident that this is what happens when you put crabs on the reef. The other interesting things were when we put these crabs on the reef and they started mauling algae off of it, we started to see these other effects. So on reefs that I did put crabs on versus those that I didn't, we started to see more fish. So I put out a bunch of cameras and did time-lapse surveys. And here, this is the blue bars are before I did anything to the reefs, and the gray bars are 1 year after I manipulated them. And you can see that the fish abundance is the number of fish in each photo that I took. And across the x-axis are the different treatment groups. So on the left is the abundance of fish, the number of fish, and on the right is the number of different species in each of those photos. So you can see in both cases, that in every treatment where I did something to the reef, where I scrubbed, where I removed algae, where I put crabs out, or where I removed algae and put crabs out, we had significantly greater species richness, so we had more species, and we had more fishes. But in the control treatments, even before and 1 year later in both experiments, there's no difference between them. These differences are driven by these 6 families of fish, and that's grunts, snappers, and wrasses, which are all predators, and then parrotfish, damsels, and surgeons that are all herbivorous fishes. Additionally, 2 years after we did these 2 experiments, we started to see we looked at just the density of living juvenile corals on the bottom. And you can see on controls versus the other 3 treatments, almost none. And then in the 2 treatments where we put crabs out, where they're physically removing algae from the reef structure through time, we had almost a 4-fold increase in what we're calling the recruitment of corals. Really, it was the survival of corals that had recruited before. So, kind of to summarize all this up, we added crabs, we reduced the cover of macroalgae, and then we saw, because of that, an increase in the richness and abundance of fish, and also the number of living corals that survived to recruit to the community. Lots of people helped me, and I got money from a few folks, and we had no questions until after.
Thank you so much, Jason. I'm going to let Dr. Mark Butler go ahead, and we'll also time him, so we still have a few more minutes for questions. So we're doing okay. 25 to 22.
So we can,
So if you could quickly plug in here, or maybe take one quick question, or maybe Mark, are you going to talk about the larval thing with the crabs, or no?
not the crabs, but lobsters.
Okay, so really quickly, maybe Jason, can you just tell everyone why, or Dave, why the crabs are so, why we're so excited about mariculture, and what is the term for it? The little eggs land in like 4 to 6 days?
Oh, they're larval durations. So many species, like Mark is going to talk about lobsters, where they spend up to 6 to 9 months with little larvae floating around in the sea. And even corals are weeks as larvae. These crabs are very interesting because their larval period is only 4 or 5 days. And then you have a crab that you can start feeding algae, will grow, and they grow rapidly. More importantly, they eat a lot of algae as little crabs, and they don't eat any more algae as big crabs than they do as little crabs. So we don't have to get them gigantic to have a measurable effect on the body.
And what's the other fancy word for the you don't have to feed the little eggs?
Oh, and they're less epitrophic. So
Say it again?
less epitrophic.
Which means you don't have to
So
feed
they
the
have-
larvae stage, right?
They have a yolk that they hatch, and they have a yolk that they feed off of, so you don't have to feed them as larvae.
So those 2 things make it very exciting that we could potentially sort of build their survival rate.
Great survival is very little.
Are there any other questions for Jason or me while Mark is setting up his video? Or talk? No?
I
Yes.
have a general question
Sure.
for the power restoration community.
Before you do that, why don't you just quickly introduce yourself, or because
I'm
it's better than me?
Mark
So,
Deputy Director, Science Advisor, so Caribbean Community Climate Change Center with IC.
thanks, yeah.
No, I just wanted to find out what role you see power restoration might play in terms of building resilience to the new emerging threat of ocean acidification.
Oh, you would asked me the hardest question, of course. I try to avoid that topic of ocean acidification because it's so very overwhelming. For those of you that don't know, that refers to the pH changing because of the CO2 being absorbed in the ocean. And I have no answer for you that would satisfy. All I can tell you is that what we've been doing these last almost 12 years now is working with what we see out there. So I know many other restorations, people like Dave, are doing experiments onshore where they're changing the pH. And they're looking, right, you're actually looking for corals that can resist or deal with lower pHs. We, as you know, Belize, we have no aquarium, no tank, no nothing. So everything we've done to date and everything that we plan to do in Belize is based on our field observations. Who's doing well, who's not doing well. But other researchers I do know are doing forced temperature and pH changes to try to figure out. Do you want to answer?
Yeah, just if anybody goes into a pet store or a reef store and says, I want to start a coral tank or a reef tank, the two things that they'll tell you is it's water quality, but you have to have a narrow range of temperature and pH. And because of climate change and putting CO2 out, it has affected both of those. One is the temperature because of the atmosphere, and the second is most of the CO2 gets absorbed into the ocean, which drops the pH. So the corals have two of the worst parts of the world. So we're going to have to see which ones are going to be resistant until hopefully mankind on this planet smartens up and stops putting all that CO2 out there. Because even if we stop tomorrow, it's going to take many years before it's all absorbed. And most of it is absorbed either in trees, on land, or it's absorbed in the ocean. So we have to pay attention to both. And we've got to keep our corals alive and functioning as an ecosystem until we turn this thing around.
Thank you for asking. Let me ask you to hold that because we only have a few more minutes. Hold it till the end, please. And please introduce yourself better than I did because, like you said, you have 3 decades of things that maybe I didn't really.
Yes, I'm old.
Couple sentences.
I'm Mark Butler.
Where is my role?
I'm Mark Butler. I'm a professor at Old Dominion University, but I've been working in the Caribbean for 35 years or so. And I've been working in Belize on and off for about 30 years. But what I really what I really like to do is just again impress upon you how special things are here because of people like Lisa. So I've been around the world, seen lots, like Dave, worked with a lot of NGOs, non-governmental agencies. And a lot of them, have a short lifespan and don't get a lot done. Best intentions don't get a lot done. I kept putting Lisa off for years to get me down here because I didn't know she was legit. And we all get a lot of demands on our time. You have demands on your time. But I finally figured out that she's legit, Fragments of Hope is legit, and they're doing some phenomenal work here. So we're going to do everything we can to help them. And it's because it's these kinds of things that really kind of get the community involved and make a big difference in the environment. So now I'm here to talk about the most beautiful of all these creatures, and that's a spiny lobster. The focus, of course, is on corals and on crabs that help corals. And there's no direct link, we can talk later on about that with lobsters, but what I've worked on for many, many years is lobster biology. And so I thought I'd talk a little bit about lobster biology, this gorgeous creature called the Caribbean spiny lobster. Now, a lot of people don't realize that spiny lobsters occur throughout the world. We always think about the claw lobsters that occur only in the North Atlantic, and there's only 3 species of those. But there's about 250 species of spiny lobsters around the world. Wherever they occur, they're valuable fisheries. And so here in the Caribbean, we have one of those species. I'm not going to talk about that. They have what we call a complex life cycle. And so unlike, say, a coral or a coral reef fish that lives its entire life in one location on a reef, spiny lobsters require multiple habitats to complete their life cycle. They start as the adults out of the reef. That's where they spawn. Their larval stage is indicative, is similar to what we see in lots of other species. That is, they go way offshore out of the open sea to complete their larval cycle. And lobsters are really phenomenal in that they have one of the longest larval cycles of any marine species. This particular species, the larvae go offshore for about 7 months. There are some species that are out there for 2 years. Somehow, and actually we know something about this, and I'll talk about it, they then go to that top stage up there we call a post-larval stage, which swims in from the open sea, comes back into the nursery habitat, and the nursery habitat is not a coral reef. The nursery habitat are vegetated areas, mangrove areas, areas that have lots of algae and some seagrasses. They live there as their juveniles, and then later move back out to the reef. So, I want to give you a little better feel for the life cycle. But it is a cycle, right? It's a circle. So, where do you start? Well, you got to start with love, right? Can you turn this up? Frank? Right? It's got to work up. The lobsters, they're not like corals that spawn. Lobsters like. Right? So lobsters have complex mating behaviors, and actually a lot of sexual selection that goes on. We've done a lot of studies about this over the years to figure out how they mate and the details of this. And any of the fishermen like to talk about it, but the key thing for a lot of our work is that big males matter. Right? Big males, they're matter. That is, if we remove all the big males, it's most of the big males that do most of the mating. And the females are choosing, they're selecting this. So there's a lot that goes on with respect to mating these animals. It's a very complex life cycle. But this is what happens then. Many of you have known lobsters. Here on the left, eggs on the bottom, those orange things. The black thing on top is the tar spot, the sperm the male transfers to her. And then she has these little eggs. And that is a larval lobster. Let's look at anything like larval. This is the stage once it hatches, it goes offshore for about 7 months. And they don't just float around. They are very actively swimming little buggers. They actually move up and down in the water column. They respond to different kinds of cues. And so it's actually a very complex process to figure out where they end up after that 7-month process. Because they're literally, and I'll show you, moving up and down in the water column large distances. But once they leave that stage, after 7 months, then they metamorphose into a stage that looks much more like a lobster. And at that stage, it finds its way back to the shore. And I'm going to show you a photo that probably no more than the people in this room have ever seen before. We reared these things for the first time. These are very difficult to rear. So we worked with folks in Japan. We reared them for the first time about 15 years ago through their entire life cycle. And we caught the metamorphosis from that stage to this other stage that normally occurs well offshore. And it's equivalent to seeing how a caterpillar goes and creates this beautiful butterfly. So we caught it in the lab, the most profound transformation in decapods. So there you can see that spidery looking thing that's laying there, but it is now molting, and you can start to see, here's this new transparent post-larval stage. It looks more lobster-like, but it's emerging from that spider-like stage that's been in the open sea for 7 to 8 months. They start in that spider-like stage, starts about the size of a pinhead, but at the end of that 7 months or so, it actually grows to be the size of about a silver dollar. So that's pretty cool to see that. Here's what those little things look like, these post larvae, and they're really remarkable because, believe it or not, that metamorphic event we just saw occurs about 25 miles offshore. And then these little things on new moon at night swim towards shore, riding the strong currents, and they're smelling their way to shore. We demonstrated they can actually detect the coastline about 20 miles offshore and swim towards the shore. And when they do, well, I'm not going to talk about this part of the research, I'm going to get to some, but here basically summarizes what happens then. So the females spawn, those are eggs, at the reef. For the first 3 months of their life cycle, those early larvae are moving up and down each day and night in the water column in the top 50 meters, top 150 feet or so. And then there's a profound change in their behavior after 3 months, and for the next 3 months, they still migrate up and down the water column during the day, but it's much deeper. It's down to about almost 150 meters, almost near say 300 feet or so. And then when they get at the shelf break about 20, 25 miles offshore, that last stage swims on shore. Now, if you wanted to predict where lobsters go to and come from as larvae, you have to understand these complex behaviors, which took us many years to figure out. And now you have to put this into complex models with genetics, because when you take this, with all the complex currents in the Caribbean, it is very difficult to determine where they go. Much harder, in fact, than predicting where they go when it's corals. And we work with Claire Paris too in Miami. She's the best in the world at doing that connectivity stuff. But corals are a cinch compared to lobsters because of the complex behaviors and the large spatial scales that we're dealing with. So lobsters can literally be all one Caribbean species, and the larvae can make it throughout the entire Caribbean. So for years we thought it was just one big mixing bowl of larvae. But now with this more complex genetics, more complex modeling work that we've done, we can now pinpoint things much better for different countries. So we can answer the question, well, there's a simple model we use. So we know now that Belize actually has two very different oceanographic regimes. Here in southern Belize, because of the currents that tend to retain things nearby, it's a retentive environment. And here in southern Belize, what we have is we get a lot of what we call self-recruitment. That is, a lot of the babies that start here come out here. So it does work like you think about along down the road, like chickens in a barnyard. However many adults you have, that's how many babies you're going to have. But that's not the case in most of the other Caribbean. And in fact, in northern Belize, it's completely different. Only about 10% of the baby lobsters produced in northern Belize never make it back to Belize. Where are they going if they're not in Belize? Well, total, about 45, approximately half of Belize's lobsters probably come back as babies after that 7 months in the open ocean. Where do they go? A lot of different places. Florida, Cuba, and actually many of them are probably lost in the North Atlantic once they get caught in the Gulf Stream. So the other half are ending up supplying Florida, Cuba, and then maybe a little bit in Bermuda, and then they're gone.
Mark, can you go back on the slide?
Yes.
Next slide. Yeah, the 90% remain in southern Belize.
Yep.
Because everything better in southern Belize.
Yeah. So they look, like I said, after they arrive here, they're looking for nursery habitats, mostly algae habitats. You can see they metamorphose, so that's still that post-larval stage, but they start getting the coloration. And then they molt again, become these little juveniles, they're about an inch long. And these little juveniles are very solitary. So at this stage, you're not going to find them aggregated. They do not hang out together. They are solitary at this stage. They don't really want to be near one another. But later on, as they start to get a little bit bigger, about this size, their whole social behavior changes. And there's some various genes that click on, and now it makes them attracted to the scent, the chemical scent of other lobsters, so they can, because when they get big enough, now being together makes them, they can defend themselves more effectively. When you're just a little one next to another one, that's just 2 bites, right? That's no protection. You got to be big enough to defend yourselves. And so they get together and they can defend themselves against predators as they get bigger and they become social, eventually going out to reefs, make these sometimes dramatic long-scale migrations offshore, and can get big, right? They can get big. Now, many people would ask me, how old is this lobster? Well, the kid's about 8 or 9. So that's a big lobster. How old? This is really hard to tell because there's a lot of things that affect growth rates. Size isn't always a good indication of age, right? For some people, there could be a teenage boy who's 13 that's 6'4", 230 pounds, and he's a lot bigger than his father might have been, 5'9". So you can't correlate size and age in a lot of things. And in crustaceans, because they molt, they shed their exoskeleton, there's no hard parts left over that allow us to age them. Almost every other thing would be easily aged. Mammals with teeth and bones, fishes that have ear bones with rings, even mollusks with shells, they have rings. Lobsters, we didn't believe had that. And so there was no direct measurement of lobster age. This just gives you, this is what you look at in a fish, a common thing we do. We can age fish because they have a little ear bone that grows rings like rings on a tree. And that's a cross section through the ear bone of a fish, and the little dots are us counting these rings. But we've discovered that in lobsters, they actually, lobsters have something, it's not like chicken is. They have hard parts in their stomachs. And it turns out those are not lost during the whole process. So we started looking, we and some other folks started looking at this, and we just literally this past year, we just published a paper, we now determined the first time that we can age lobsters using these hard parts in their stomachs. It's a complex process of how we do this, but there is one of these pieces from a lobster's stomach. You can clearly see the bands, and those dots are how we can actually age lobsters using this. So we're still in the middle of this process, but it looks like it works pretty well. So we were very, very fortunate that in Florida we had colleagues in the state of Florida that had been raising lobsters in the lab from that tiny little post-larval stage for 10 years. 10 years. So nobody has no-age animals like that. Nobody keeps a lobster and grows it for 10 years like that. But they did this because they hoped some method like this would come along. Now there might be some wild artifacts, but we use this method so we can now correlate what the age and size we see. There might be a little bit off here, but we're doing more field experiments to look at this. Believe me, it was very, very hard to get those 10-year-old lobsters. I had to go to my friend and say, can I kill your dog? It's like, right? They had names. These were big lobsters. He grew them for 10 years. And we had to kill them to get this information. But, so how big is that? How old is that big lobster? Well, that's our best guess. That big lobster probably grew that big in about 7 or 8 years. We previously thought it must have taken about 25 years to get that big, but we now think that they can grow that big much more rapidly. I'm going to make a quick couple things. There are some fishermen here? Yes? No? Lobster fishermen? Yeah, a few of you. So you probably have heard about the lobster disease that's occurred. It was my lab that discovered this in Florida. We've since gone around the Caribbean to document this. So this is basically AIDS for lobsters. Literally, it's a viral disease. It's transferred by communication, by close contact among lobsters. Those are the actual viruses on the right-hand side. I'm not going to go into the details, but I'd be happy to tell you guys, talk for fishermen later about this. We discovered it in 1999 in Florida. It's the only known viral disease in any species of lobster anywhere in the world. It's a unique new virus for lobsters. It's not something that's recent. I mean, I've been studying lobsters for 35 years. We saw this disease 35 years ago. We just didn't know what it was. So it wasn't that the virus just happened in 1999. It's been around. And the problem is, the adults you don't see it so much. It's much like a lot of human disease. It's the young ones that can get this disease more easily, and it's lethal. This is worse than Ebola in terms of its effect. Ebola kills about 60 or 70% of people infected. This stuff, the juveniles, 90% of them are killed by this stuff. If they make it past that, they're fine. So we estimate that in Florida, for example, we're probably losing about 1/4 of the fisheries probably being lost to the little babies that are getting this disease, so the fishermen never ever see it. They're killing the babies before you ever see it with this virus. It doesn't have any other known hosts, so it's very specific to lobsters. What does it look like? Some of you fishermen have probably seen this with some of the smaller animals. When you see this kind of milky white blood that they get sometimes, and sometimes they'll get this off color, a little bit kind of reddish more. Those are pretty good, visual signs. But to be honest, you can't. That's a lobster that's going to die within a week. That's very late stage. To really determine whether they have the virus or not, we actually have to draw blood. We can take it to the lab, analyze it. Here you can see, here's a healthy lobster on the left of this. If you look in that little window, that diamond-shaped window, you can see the clear blood, and you can see on the right, it gets that milky white kind of color. So, but that's only good for the late stages. We really have to draw the blood, send it to a lab, and get it detected for the virus. But here's just an idea of the prevalence around the Caribbean. Just about 10 years ago now. White means areas that didn't have the virus. Yellows, there are some virus. Red areas are areas where there's a lot of this virus in the population. So we have not been able to update this. Why? Because it costs money to do this, and getting grant money is hard to do. So we haven't updated this in about 6 or 8 years, it looks like. One of the real interesting things about this, and almost is that, believe it or not, lobsters have evolved with this virus and have evolved a really remarkable behavior. The first behavior that's ever been shown in nature in this, and that is a healthy lobster can chemically detect there's an infected lobster, but you can detect that chemically before it ever is transmissible. So it's like, I know you're going to have a cold next week because the virus is in your system, and I'm not going to shake your hand because I know you've got the virus. So what do? This normally social species stays away, quarantines the disease ones. So the disease ones tend in nature to be found alone. One thing that's important for fishers to understand, though, doesn't seem to happen if you see this or shades because they get separated. But if you have lobster, if you have a diseased lobster in a trap, it can transfer the virus to other lobsters in the trap. So if you're a lobsterman, what do you do with those diseased lobsters? Take them, do not throw them back in the sea. That virus leaks out in the sea. Take them back with you and dispose of them on land. That's about the only thing we can do because we can't inoculate the whole ocean with some kind of antivirus. So with that, thanks for sticking around and talking about lobsters. In terms of staying, it's all hydrodynamics. It's the sea, right? It's the sea. When you get to northern Belize, it's a different oceanographic. The water in northern Belize shunts up to Mexico, and that's where the Gulf Stream kind of begins. And southern Belize and the Gulf of Honduras, so southern Belize, Honduras, Guatemala, those populations are very much interconnected because of currents. It's possible, yeah. It's possible.
My big question was, you said, so the biggest risk for populations in Belize is temperature rising, precipitation? No, I did not say that's the biggest risk. We could, if we wanted to say that climate change, which encompasses those things and more, is probably the biggest risk. But when we also talk about pollution, wastewater runoff, sewage, sunscreen. So, I saw some debates recently about people saying, what is the worst threat? It's easy to say climate change is the worst threat, but at the same time, there's things we can do right now, like stop with the sunscreen, work on our wastewater mitigation, that are a little bit more in grasp than suddenly everybody stops emitting fossil fuels.
Sorry.
So I have a lot of public members who are lobsters, but this is the first time that I'm hearing that lobsters do have a virus, can carry a virus, and I would like to know how does the virus affect the lobster, like in terms of growth, and how does it affect us as humans if we would eat the lobster?
Right, yeah, that's a good question. So the question was, how does this virus affect lobsters, and can humans get it? The answer to the second part is no. You cannot get it as a human. So, but the lobsters, again, it's lethal. So this virus basically first gets into their blood cells, basically disintegrates their blood cells. If you ate it, it's not going to do anything to you. It won't affect you.
It's a good way to dispose of it.
Yeah, well, if you want, but I want those ones that milky white blood, you can eat it, dude, not me. That is nasty.
All the crazy one,
You go right ahead, man. I'll send it to you. Within a month. Within a month, they will die. It's about,
I need to lose.
it's around 20% in Belize. When we sampled in Belize, I sent to about 6 or 8 different places in Belize, took blood samples, went back, and that's what the results. Not a little, littles are very hard, so. Oh, for years. For years, yeah. The early stages you can't tell. So you would never ever know. it's not going to kill you. It has no.
Any other questions? Anybody for the speakers? Okay, people, thank you so much for coming out. People in our course, do not show up to my door. Especially if you need to hear.
Thanks, Mark. That was great. So we're going to log off now. That was Fragments of Hope, Corals and Crab, live from Placencia, Belize.